Dear students, in the last modules, we studied about route of administration of drugs and the dose-response relationship. After studying this module, you shall be able to know about the significance of alkaloids, classification of alkaloids and importance of some notable alkaloids, and forensic importance and examination of alkaloids. Alkaloids are a group of molecules with a comparatively large occurrence in nature around the world. They are very diverse chemicals and biomolecules, but they are completely secondary compounds and they are derivative of amino acids. However, an exact definition of the word alkaloid, that is alkali-like, is slightly difficult because there is no clear-cut borderline between alkaloids and naturally occurring complex amines. The term alkaloid was coined in 1819 by the pharmacist W. Misner and meant simply alkali-like. Typical alkaloids are derived from plant sources. They are basic, they comprehend one or more nitrogen atoms generally existing in a heterocyclic ring, and they habitually have a noticeable physiological action on human or other animals. The term Proto Alkaloid or amino alkaloid is occasionally applied to compounds such as codeine, ephedrine and colchicine, which lack one or more of the properties of typical alkaloids. Alkaloids are organic bases. A true alkaloid can be regarded as by the possession of a nitrogen atom as part of a heterocyclic system, a multifaceted molecular structure, significant pharmacological properties, and its origin from the plant kingdom. The majority of alkaloids fit this four-part definition. However, a number of exceptions exist. The compounds samidorine, samidrone and cycloneosamidrine, secluded from the dermal glands of the European fire salamander, displays the usual properties of an alkaloid substance, but do not fit the definition of a true alkaloid owing to their animal origin. True alkaloids are of rare occurrence in lower plants. In the fungi, the lysergic acid derivatives and the sulfur-containing alkaloids, example, the gliotoxins, are the best known. Many alkaloids are poisonous, others are addictive, example cocaine, and some are used clinically, example morphine. More than 10,000 alkaloids are currently known, the first discovered being nicotine isolated from opium. Alkaloids are constituents very well recognized for their biological action at the commencement of world evolution. They were used in traditional herbal medication for the treatment of ailments and in weapons as venoms in the course of tribal wars and during hunting. After the discovery of the first alkaloid, nicotine, other alkaloids were rapidly discovered like morphine from opium, strychnine from Strychnus nux vomica seeds, amytine from Ipecacuanha root, and quinine from cinchona bark, and quinine from hemlock. Alkaloids are used medicinally in their own right, example, hyoscyamine and vinblastine, and they have often provided lead compounds for the development of synthetic drugs. For example, cocaine was the first local anesthetic, quinine first antimalarial, and tubocurarine the first neuromuscular blocking agent. Alkaloids frequently have powerful physiological effects. Most alkaloids are colorless. Crystalline compounds, example conines, but some are nicotine and high green are liquids. Usually, Many are optically active in nature and the various active forms are generally found but not in different plants. Alkaloids are produced by secondary metabolism of primary metabolites, usually amino acids. These pathways are long, intricate, stereochemically precise and energy consuming and are assumed to be of evolutionary benefit. Forensic significance of alkaloids. The poisonous and therapeutic effects of plants have been known since time immemorial, but the active constituents have been studied for only about 200 years. In most human history, alkaloids from plant extracts have been used as ingredients in potions, that is, liquid medicines and poisons. Ancient people used plant excerpts containing alkaloids for the treatment of a large number of diseases such as snake bite, fever and insanity. Habitually, the bioactive compounds in the plants that intrigue the chemist are the nitrogen-containing bases called alkaloids. Alkaloids have complex molecular structure and they have significant pharmacologic activities. Alkaloids are time and again toxic to man and have many intense physiological actions, hence they are widely used in medicine. For examples, cinchona alkaloids, which is existing in the bark of cinchona species, and remigia species consisted of quinine as their main constituents, which has been known as anti-malarial agent. Alkaloids have delivered a means of pharmacologically active compounds. Approximately 25% of the drugs used at present are of plant origin. These are administered either as pure compounds or as extracts and have often served as model structures for synthetic drugs. Next is chemical properties of alkaloids. Maximum alkaloids are well-defined crystalline substances which bond with acids to form salts. In the plant, they may exist in the free state as salts or as N-oxides. In addition to the elements carbon, hydrogen and nitrogen, most alkaloids contain oxygen. A small number of alkaloids such as quinine from hemlock and nicotine from tobacco are oxygen free and are liquids. Even if colored alkaloids are comparatively uncommon, they are not unknown. Berberine, for example, is yellow and the salts of sanguinarine are copper red. Next is classification of alkaloids. Classification based upon the biogenesis. Alkaloids show great variety in their botanical and biochemical origin, in chemical structure and in pharmacological action. Consequently, many different systems of classification are possible and the classification mentioned hereinafter is not exhaustive. Alkaloids can be classified by their mutual molecular precursors founded on the biological pathway used to build the molecule. From a structural perspective, alkaloids are separated according to their shapes and origins. There are three main types of alkaloids. First, true, second, proto alkaloids, and third, pseudo alkaloids. True alkaloids and proto alkaloids are derived from amino acids, whereas pseudo alkaloids are not derived from these compounds. First, the true alkaloids. True alkaloids are derived from amino acids and they share a heterocyclic ring with nitrogen. These alkaloids are extremely reactive substances with biological activity even in small doses. All true alkaloids have a bitter taste and appear as a white solid, with the exception of nicotine, which is a brown liquid. True alkaloids form water-soluble salts. Furthermore, most of them are crystalline substances which bond with acids to form salts. True alkaloids may occur in plants in the free state as salts and as N-oxides. These alkaloids befall in a narrow sum of species and families and are those compounds in which decarboxylated amino acids are condensed with a non-nitrogenous structural moiety. They are typically found as salts in plants such as lirodinine and morphine. Second are Proto Alkaloids. Proto Alkaloids are compounds in which the nitrogen atom derived from an amino acid is not a fragment of the heterocyclic ring. Such kinds of alkaloid include compounds derived from one tyrosine and one tryptophan. Proto Alkaloids are of a closed ring, being impeccable but structurally simple alkaloid. They are not restricted to any specific class of alkaloids and are often classified according to the amino acids from which they are derived. They form a minority of all alkaloids. Codeine, mescaline and yohimbine are good examples of these kinds of alkaloids. Third is Pseudo Alkaloids. Pseudo Alkaloids are compounds of elementary carbon skeletons of which are not derived from amino acids. They can also result from the amination and transamination reactions of the different pathways coupled with precursors or postcursors of amino acids. These alkaloids can also be derived from non-amino acid precursors. The nitrogen atom is introduced into the molecule at a relatively late stage. For example, in the case of steroidal or terpenoid skeletons. Pseudo alkaloids can be acetate and phenylaniline, derived or terpenoid as well as steroidal alkaloids. Examples of pseudo alkaloids includes such compounds as quinine, capsaicin, ephedrine, solanidine, caffeine, theobromine and pyridine. Next is the classification based upon the ring system. It is perhaps the most extensively established and mutual approach of classification of alkaloids, for which the principal condition is the presence of the basic heterocyclic nucleus. According to this concept, the alkaloids are classified on the basis of the ring system present in them. First is pyrrolidine alkaloids. Pyrrolidine alkaloids have a pyrrolidine nucleus, that is C4N skeleton. The structural alpha of these alkaloids is 1 ornithine in plants and 1 arginine in animals. Example is hygrine. Second is piperidine alkaloids. Piperidine alkaloids contain the piperidine nucleus. True piperidine alkaloids have 1 cycle compounds with the C5N nucleus. One of the characteristics of piperidine alkaloids is the odor. Moreover, they are neurotoxins which have acute effects such as chronic toxicity. Example is lobelia. Third is pyrolizidine alkaloids. The pyrolizidine nucleus is characteristic of this group of alkaloids. Example is cinchona. Next group is tropane alkaloids. Tropane alkaloids have a tropane nucleus, that is C4N skeleton. Structurally, these alkaloids synthesize as postcursors of pyrrolines. Characteristic tropane alkaloids, example atropine, hyoscyamine, cocaine, troponon, tropine, litorine and cuscohygrine have a strong biological activity, especially as neurotransmitters. Next group is quinoline alkaloids. The alkaloids containing principally the quinoline nucleus comprises a series of alkaloids obtained exclusively from the cinchona bark. These alkaloids possess the basic skeleton of 9-rubenol, that is derived from the parent compound known as ruben. The major members of this particular group are namely quinine, quinidine, cinchona and codeine. Next is isoquinoline alkaloids. The isoquinoline alkaloids are a huge class of medicinally active alkaloids whose properties are variable. Their properties include being antispasmodic, antimicrobial, anti-tumor, antifungal, anti-inflammatory, antiviral, amoebicidal and antioxidant, and can also act as enzyme inhibitors. This class notably includes morphine and codeine. They are derived from the amino acids phenylalanine or tyrosine. They are formed from a precursor of 3,4-dihydroxytyramine, that is dopamine, linked to an aldehyde or ketone. Examples: opium alkaloids like piperidine, nicotine and narcine, pancreatistatin, sanguinarine, hydrastine, berberine, amytine, buramine and oxyhexanthine. Next group is indole alkaloids. This structural group of indole alkaloids contains indole ring. Example: tryptamine, serotonin, ergometrine, xylosine and psilocybin. Indole alkaloids form a very important group from the perspective of their application. Examples: tryptamines, serotonin, bufotenin and psilocybin. Ergolines, ergine, ergotamine lysergic acid. Beta carbolines, examples: harmine, harmaline, tetrahydroharmine. Yomibands, examples: rasparine, yohimbine. Vinca alkaloids, vinblastine and vincristine. Kratom alkaloids, mitragine and 7-hydroxymitragynine. Next is terbinanthe iboga, ibogaine, vocaine and coronaridine. Next is strychnus nux vomica, strychnine and brucine. Eighth is imidazole alkaloids. Imidazole alkaloids contain an imidazole ring structure in them. This group of alkaloids is an exception in the transformation process of structures because the imidazole nucleus is already made at the stage of the precursor. Alkaloids from this group include, for example, dolichocylene, pilocarpine and pilocene. Next is purine alkaloids. The coffee plant family contains two major purines of adenine or guanine derived alkaloids, the so-called purine alkaloids. Purine is a nitrogenous base of nucleotide which consists of just purine and pentose sugar, that is D-ribose or 2-deoxy-D-ribose. Typical purine alkaloids are caffeine, theophylline and theobromine. The same or similar purine alkaloids occur also in other plant families. Next is steroidal alkaloids. Steroidal alkaloids ascend by the presence of basic nitrogen at some point in the steroid molecule. Example, solanidine. Some notable alkaloids of forensic importance. First is ephedrine. Various species of ephedra are used as a source of the alkaloids ephedrine and pseudoephedrine. Ephedrine is used for the relief of asthma and hay fever. Its action is more protracted than that of adrenaline, and it has the additional advantage that it need not be given by injection but may be administered by mouth. In Oriental medicines, the ephedras are also used as anti-inflammatory drugs, and this action is attributed to an oxalidione related to ephedrine. Ephedrine may be applied in the treatment of edema in insulin-dependent diabetics. Next is quinine. Quinine was the first alkaloid to have its structure recognized and to be synthesized. Piperidine alkaloids such as quinine and quinacine are very poisonous. They occur in hemlock, Conium maculatum, known as a very toxic plant. Cases of death by respiratory failure resulting from quinine alkaloids are frequently reported. The classic toxic symptoms of quinine alkaloids range from paralysis, muscular tremors, muscle weakness and respiratory failure preceding death. When hemlock is treated with solution or potassium hydroxide, it develops a strong mouse-like odor owing to liberation of the alkaloid quinine. Next is nicotine. Nicotine is a significant insecticide and large quantities of it are prepared from those parts of the tobacco plant which cannot be used for tobacco manufacture. Nicotinic acid provides alkaloids with the pyridine nucleus in the synthesizing process. This nucleus appears in such alkaloids as anabasine, anatabine, nicotine, nornicotine, ricin and acetylenine. Nicotine is used in countless products on the pharmaceutical market. At least 20 different products are known to contain nicotine. Next is atropine. A chief source of atropine is from the plant Atropa belladonna. Atropine has a stimulant action on the central nervous system and depresses the nerve endings to the secretory glands and plain muscle. Atropine blocks muscarinic cholinergic receptor competitively and has a large spectrum of clinical applications. Atropine acts as a parasympatholytic or parasympathetically innervated organs. Therefore, the possible applications of atropine are as a general anesthetic and include its use in pure form or as a component. Next is quinine. The pharmaceutical drug industry has succeeded in the use of natural plant alkaloids for the development of antimalarial agents like quinine. It is acquired from the bark of cinchona plant. Also because the nucleic acid synthesis is inhibited by the DNA intercalation, quinine has a schizoneticide activity. This activity is also based on carbohydrate metabolism. Quinine can be used in the treatment of myotonic disorders. Quinine can be found in drugs such as Adaquin or Biquinate. These drugs are important in the prevention and treatment of malaria. Next is cinchonin. Cinchonin is obtained from a variety of cinchona bark, especially in the bark of Cinchona micrantha, belonging to family Rubiaceae. The basic skeleton of cinchona alkaloids is Reuben 9-ol. Reuben nucleus is a combined skeleton formed from a quinoline ring attached through a methylene group to a quinucleotide ring, a bicyclic ring containing nitrogen. Cinchonin and cinchonidine are used as an anti-rheumatic. Next is morphine. The plant family Pepiveraceae contains 1-triosine derived alkaloids such as morphine, codeine, thebaine, piperine, narcotine, etc. The opium poppy, that is Papiverum somniferum, is a well-known source of opium from its latex. The poppy family alkaloids have strong biological and medicinal impact. They are also strong narcotics. The morphine molecule has both a phenolic and an alcoholic hydroxyl group, and when acetylated forms diacetyl morphine or heroin. Next is codeine. Codeine is ether of morphine, that is methyl morphine, and other morphine ethers which are used medicinally are ethyl morphine and pholcodeine. Codeine is a constituent of at least 250 pharmaceutical products in the market. Next is strychnine. Strychnine, obtained from the plant Strychnus nux vomica, is an extremely toxic alkaloid. Glycine gets accommodated when strychnine binds itself to receptor sites in the spinal. Brucine is a dimethoxy form of strychnine and is less toxic. Strychnine may also cause convulsions and disorders of the central nervous system. We'll end this module with the summary. The alkaloids are defined as the basic nitrogenous complex organic compounds available naturally in the plant and animals. Generally, alkaloids contains a heterocyclic ring in their molecular structure, but non-heterocyclic structures have also been reported. The very first technique employed for the analysis of alkaloid was developed in the year 1805 for morphine. Morphine and codeine are natural products of Papaver somnifera. However, the codeine is naturally produced in small amounts. Alkaloids are found essentially in the plants of the dicotyledonous families. They occur in certain parts of the plants like leaves, fruits, seeds, bark and roots. Most of the alkaloids have been colorless except berberine, crystalline, non-volatile solids except quinine and nicotine, and are bitter in taste. Alkaloids are basic and go through reaction with acids to form alkaloidal salts. The alkaloids are unbound from their salts by adding alkalis. Morphine and codeine nitrogen oxides are natural constituents of the opium poppy latex, and Nicotiana species contain 2 isomeric nicotine N-oxides based on the pyrolidine nitrogen. Cocaine was the first local anesthetic and quinine was the first, and tubocuranine the first neuromuscular blocking agent. Alkaloids can be precipitated by the reagents: Mayer's reagent, Dragendorf's reagent, Hager's reagent, Wagner's reagent, Mermi's reagent, Somichin's reagent, Skibler's reagent, and tannic acid solution.